My name is Tim Szewczyk. I'm a lecturer in ecological modeling at the Scottish Association for Marine Science, and I'll talk today about some work I've been doing with sea lice in the context of salmon aquaculture. So salmon farming is a big business in the North Atlantic, especially in countries like Norway and Scotland, and the number of farmed salmon actually outweigh the number of wild salmon by an order of magnitude or more. And one of the biggest challenges that they face in terms of sustainability is sea lice. So these are external parasites that feed on the kind of skin and mucus of the fish. So they cause a lot of health issues for the fish themselves, costs for management, and they also The interesting thing about the sea lice life cycle, if maybe you're used to thinking about diseases or parasites that maybe you're more familiar with, like say ticks, is that sea lice have this larval stage that they're planktonic. So they after they hatch, they go through what's called the nauplius stage. It's actually 2 stages. During that time, they're not feeding. And then after 3 to 10 days, depending on the temperature, they molt into copepodids. And it's this stage that can then find a host and attach. But during this whole time, they're mostly being transported around by ocean currents. So after this copepodid then attaches to a fish, it molts through a few additional successive stages, and then finally into an adult, at which point they're reproductively active. And that process takes on the order of weeks. So if we're concerned about controlling infestations, predicting risk to wild fish, it's really this copepodid stage that we're most concerned with. The trouble is that they are very, very small. They're sparse and patchily distributed, so you have to sample like 10 cubic meters of water to even have a good chance of finding one. And then it takes a trained taxonomist half a day to a day to sort through that sample to find the maybe one sea louse that's amidst all that zooplankton. So instead, we're left dealing with the largely industry-based counts of lice on their fish and modeling to fill the gaps. For the larvae, we use particle tracking models generally. So in this modeling structure, we're simulating the movement of particles through a model of the ocean, so ocean currents and temperature, salinity, and we can assign various kinds of biological attributes to these particles. So we can give them development rates, mortality rates, even vertical behavior, so they swim up or down in response to environmental triggers, which then affects which currents they have access to that are flowing in different directions. As you might expect, these models can get very complicated, and the truth is that there's a lot of uncertainty with all of these different choices. So here, these are 2 different example simulations with the same time, same starting conditions, but with different sea lice behaviors. And so any one of them, either one of these would be plausible given what we know. It would pass peer review and scrutiny by the regulators, but you'd come up with very different predictions for the density, the distribution of these sea lice. And given the limited direct observations of larvae that we have, it can be a big challenge to actually validate these models in any way, in any meaningful way, and to try to tune them. So what we have to do is take those outputs from those models and try to align them with the observations that we do have at these later stages. So that means you have to account for this difference in timing of generally weeks between when the larvae attach and when they're actually observed, as well as all of these population dynamics that occur. To make matters worse, any observations we have of the early stages tend to be very error-prone as well because they are still very small and difficult to detect. So some of us have been exploring the idea of using ensembles to try to characterize some of the uncertainty here. So it's kind of probably to everyone here an intuitive first step to take is that if you have a lot of uncertainty in these simulations, just run a few of them and at least capture the range of outcomes that are reasonable. And it turns out that if you run a set of simulations, and then if you have appropriate data to calculate a weighted mean, those weighted means, those models perform better than the best of their constituents, right? So you can actually outperform any of your individual simulations. And even if you don't have that data, a simple average of the time series from these simulations will perform better than the median. And since did this in STAN, with the Bayesian model, you have the added upside that you're also characterizing the uncertainty when you're making these kinds of predictions. This particular study was a bit more proof of concept, and so all of that biology, that complicated biology, was treated in a very basic and crude way, and we only used adult lice. So what I've been working on since this was dealing with that gap. So here, this is my current modeling system, I guess, for sea lice. So it's split up into two pieces. We have this ensemble of dispersal models. So we run a set of them with different parameterizations. And then we take those outputs and join them into an estimate of the ensemble infestation pressure, the ensemble number of copepodids, this infectious stage in the water. Then we model attachment, the progression through the stages, and then ultimately align that with the observation of fish. Right, so we have the modeled number of larvae at an hourly scale at each location of interest, and then we have typically about weekly counts of the mean lice per fish on these farms. So for the model I'll show you here, it's across 3 different years. Here we are in Western Scotland. That's my house there. And we had, from our industry partner, detailed data from 10 different farms across these 3 years. Those are the farms in yellow there. So we have the full population model that we perform for those sites, but then the dispersal modeling takes into account all of the different farms within the region. So they're kind of all producing larvae for these simulations. I won't say much about the actual dispersal simulations, but suffice to say that there were 10 of them. Each of them would be plausible, and they're designed to be fairly distinct from one another. And each of them outputs this hourly prediction for the number of infectious larvae per cubic meter at each farm across those 3 years. For the STAN model, I'll kind of step through this piece by piece. In my head, I break it up into 3 different components. The first deals with trying to estimate what number of lice actually attach to the fish. Right? So we go from this time series, these time series of predictions from our particle tracking simulations through to how many have actually attached. So the first step there is to just estimate these weights. I won't go into detail there. Happy to talk about that later. I've tried out a few different ways of dealing with this step with varying levels of complexity. The current iteration, it's just static weights for these 10 different models. We also include background infestation pressure. So the idea here is that these are larvae that are produced by wild populations, and we allow that to vary seasonally. So we use harmonic terms for the day of the year to capture those background processes. And then we estimate the attachment probability. So this is all done on an hourly scale because we have environmental conditions that are fluctuating and copepodid distributions that are fluctuating rapidly. And so this attachment rate is a function of how many hosts there actually are, so like the total surface area of fish on that farm, the salinity, temperature, and current speed. So we do a little bit of arithmetic, and we get from those hourly estimates to daily averages. And since this is a population model, we only really care about the females. The next part of the model progresses the populations through these stages. So this is a fairly standard approach in population ecology. We just use a form of a matrix model. The only difference that's a bit unique is that we estimate daily rates for everything. So we have a daily survival rate for each stage that depends on the salinity. We also need to deal with anti-lice treatments that are applied periodically by these farms. So we have an estimate for the efficacy of those treatments, basically extra mortality on the days when they apply some kind of treatment to try to kill the lice. For development, it's a function of temperature, and that is parameterized in terms of the mean stage length, so the mean number of days per stage. And so we take the inverse of that, and that gives us the proportion of individuals in that stage we would expect to progress to the next stage each day. And then we just take those values and compose them into our projection matrix, which allows us to take the population vector from one day, multiply that by that matrix, and then calculate the population vector for the next day. And lastly, we add in the new number that have attached. So then we have our latent expectation there for the mean number of lice per fish in each of these stages, each day on each farm. Then we need to deal with how they're actually sampled. So here, the industry, the farms capture some number of fish. It's between 25 and 400 on the day that they sample. They count all of the lice on those fish, and then they only track the total number of fish that they've sampled and the total number of lice. To make things slightly more complicated, they also don't distinguish all 5 of these stages. They just lump together Chalamus 1, Chalamus 2, pre-adult 1, pre-adult 2. So we just add those together, scale by the number of fish that have sampled, and then also account for detection error. We assume that the adults are all, any adult that's present has been counted, but for these earlier stages, we have a detection error associated with them. And then lastly, we have the observations distributed following a negative binomial distribution. So that's the whole model. In terms of priors, there's varying degrees of evidence. It's for attachment in particular, everything is a bit circumstantial in my opinion. But for survival, we're much more confident that it's low salinity that has, if any impact at all, it's going to decrease survival. So we've worked in empirical data where we have it. The model is still a work in progress. I was optimistic that it would be fully finished by this week, but it's not quite there. It's doing an okay job, but we still have a bit of a mismatch with the distribution here. So still working this out. I think we have an issue with a bit of zero inflation, where we have more and more zeros than we would expect, and that's kind of pulling a lot of these predictions downward. But from this model, we can estimate some really useful things that are otherwise not available. So, for example, on each farm, we can predict not just the number of infectious copepodids we would expect to be there on a given day or given hour, but we can actually predict how many we would expect that would actually attach, given the conditions related to attachment. And with all of this, because it's calculated from the posteriors, we get full posterior distributions for all of these, so it includes uncertainty. We have some grants under review right now that would actually work this into a bit of a forecast. So the hydrodynamic models we're using to run these simulations have a 5-day forecast associated with them, so we could actually predict out ahead. We could also do things like project out to a full map, potentially, to get better estimates for the risk to wild fish. For the attachment rates, this is showing the kind of tentative results here. So we do see that attachment tends to be lower at high temperatures, and also that attachment's higher when you have more fish. We didn't find much in the way of currents or salinity. And similarly with survival, this matches pretty well with the domain expectations from domain knowledge, that it's the earlier stages that tend to be more vulnerable to low salinity. So one of the anti-lice treatments is actually fresh water, and that has a pretty major impact on these early stages, not much impact on the adults. So the model is recovering these things that weren't put into it, but match with what we know about the system. So, like I said, it's still a work in progress, but to my mind, Bayesian modeling seems like a very obvious solution to a lot of the problems that we face in this system in particular, but in ecology in general, honestly. So here we're dealing with a very complex system with a lot of uncertainty, a lot of natural variability, and the current state of the art, I guess, is to try to tune your dispersal model as best you can, and just use that one dispersal model. There's kind of ad hoc comparisons, but I think it's much better to recognize that these are systems with a lot of uncertainty, and that we're talking about populations with millions of sea lice. And so the predictions you're going to get if you just have kind of the mean behavior or mean parameters is going to be different than the map you get if you incorporate all that variability. On top of that, then we have these limitations in terms of our data. So I've been working on this, like I said, to ultimately try to make some kind of more or less real-time system, a predictive system, to aid in the farms in their decision-making and to make regulation a bit more robust in the way that it treats the uncertainty within this. So with that, thanks to all of my collaborators, funders, and thanks for listening.
Great. Thanks so much, Tim. I think we'll stick to just one question for Tim, if that's okay. Anybody? behind you over there.
Thanks, that was really interesting. I'm just wondering, your model is not spatially explicit, it's spatially implicit through the particle tracking. So I just wondered whether you had a sense of how much, how much there is an effect of farms being close to each other, whether there's an opportunity to have more explicit sort of spatial component into the model.
Yeah. So like you said, some of that is implicit within the dispersal modeling. So in the paper that has been published, that actually covered the whole west coast of Scotland. And so for that, the ensemble weights were actually a function of of space. So it had some kind of spatial smoothers, some GAMs that vary by latitude and longitude. The expectation there is that the dispersal model might work better under the hydrodynamic conditions in one lock versus in another. So that's something that I have included. I haven't actually gotten to the point where I've done that kind of analysis on this particular model. But yeah, I mean the connection between them should be captured by the particle tracking model and the environmental conditions in theory. Yeah.
Thanks.
Super. Hardly an objective observer, but I thought all three of these talks were just fantastic and clear. So please join me in thanking Anna and Colin and Tim. Great. Sarah, I don't see you. Do you have any?